Hello everyone, my name is Katie Khang and I'll be discussing the role of NRF2 in lipopolysaccharide-induced inflammation in mice heart tissues. To begin, I want to introduce some of the subject matter I'll be discussing today, such as sepsis. So sepsis is a whole body immune response to infection or injury of the body due to bacterial and viral infections, and each year over 200,000 people die from it. LPS is an endotoxin that is part of most gram-negative bacteria, which can trigger the production of inflammatory cytokines, resulting in sepsis. The production of these cytokines can lead to tissue inflammation, resulting in septic shock. The image I'm pointing to shows a pathway of how LPS can induce inflammation, leading to sepsis. So LPS is directly attached to the wall of gram-negative bacteria and can trigger the activation of host immune systems using toll receptors. These receptors will then trigger the activation of the pathway nuclear factor kappa B, which leads to the production of pro-inflammatory cytokines. The image to the right in the middle I'm pointing to shows a pathway of NRF2. NRF2 is a transcription factor that regulates the expression of detoxifying enzymes and antioxidant genes that protect cells from various injuries via their anti-inflammatory effects. NRF2 reduces inflammatory damage by regulating these inflammatory factors such as pro-inflammatory and anti-inflammatory cytokines. Now, D3T is a common dithion known for its impact as a cancer chemopreventative agent and its anti-inflammatory properties, but it remains unclear whether D3T can reduce LPS-induced inflammation in mice heart tissues. In this study, I investigate LPS-induced inflammation in heart tissues from C57B6 mice and if D3T can mitigate LPS's inflammation and its underlying mechanisms. To accomplish this, I obtained multiple samples of heart tissue from NRF2 wild-type and NRF2 knockout mice. The mice were treated with either LPS, D3T, or LPS and D3T, or no treatment at all. The image to the right I'm pointing to is a short example of the steps of RNA isolation I performed on the samples. RNA isolation, I measured the RNA concentration and purity with a nanodrop machine. The image below I'm pointing to is an ideal model of a nanodrop measurement. I then performed cDNA synthesis with the acquired RNA samples, and lastly, I performed quantitative real-time PCR with the cDNA samples. Here, Figure 1 shows the results of amplification plot after performing real-time quantitative PCR on two different samples with multiple genes. Now, Figure 2 shows the results of a melting curve after performing RT-qPCR on two different samples with multiple genes as well. Figure 3 shows the amount of NRF2 anti-inflammatory gene expression of GLC in wild-type and knockout mice. In graph A, there's a significant decrease in the gene expression of LPS treatment when compared to the control, and there's a significant increase of the gene expression of LPS and D3T treatment when compared to LPS. In graph B, there is no significant change, meaning that NRF2 plays some role in the effectiveness of D3T's anti-inflammatory effects. Now, Figure 4 shows the amount of GST anti-inflammatory gene expression in NRF2 wild-type and knockout mice. In graph A, there's a significant increase in the gene expression of GST when treated with LPS and D3T. And in graph B, there seems to be no significant effect. Lastly, graph A shows the amount of NQO1 anti-inflammatory gene expression in NRF2 wild-type and knockout mice. As we can see, there's a significant decrease in the gene expression of LPS treatment when compared to the control in graph A. There's also a significant increase of the gene expression when treated with LPS and D3T treatment when compared to the LPS in graph A. Now, graph B shows no significant effects. And lastly, Figure 6 shows the amount of IL-6 pro-inflammatory gene expression in NRF2 wild-type and knockout mice. There's a significant increase in the gene expression when treated with LPS, and a significant decrease in the gene expression when treated with LPS and D3T in graph A. In graph B, there's no significant change, meaning that NRF2 plays some role in the effectiveness of D3T's anti-inflammatory effects. As a summary of my results, in NRF2 wild-type mice, D3T inhibits LPS-induced expression of IL-6 pro-inflammatory genes. In NRF2 wild-type mice, LPS markedly reduces the expression of NRF2, GST, NQO1, GST anti-inflammatory genes, and D3T is able to ameliorate LPS's responsiveness. LPS-induced inflammation and the protective effect of D3T against LPS was diminished in NRF2 knockout mice, suggesting that NRF2 plays an important role in regulating D3T's action. This study would increase the understanding of the role of D3T on LPS-induced inflammatory injury.